feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, and welcome to the National University of Singapore Yonglu Lin School of Medicine Healthy Longevity webinar. Thanks again for joining us tonight. We have a great show with Stuart Cook, and I want to remind you to use the Q&A function to ask questions, and we have Kalisha here to take those questions. One comment quickly is that I'm here in my living conditions in my condo at the NUS faculty housing, and the air conditioner quit working a few minutes ago. So if I'm sweating like Donald Trump would if he actually had to testify at one of his indictments, it's not because I have anything to hide. It's because it's really hot and humid here. So I'm going to do my best. Tonight we have Anderson, who's a research assistant in the Healthy Longevity Translational Research Program here at NUS, talking to us about joint associations of physical activity and sleep deprivation during cognitive aging.
Thank you for a kind introduction. Hi everyone. As mentioned, my name is Anderson, and today I'll be sharing some recent research insights on the joint association of physical activity and sleep duration on cognitive aging with you. This article was published by Bloomberg and colleagues in the journal Lancet Healthy Longevity. Data used in the analysis was obtained from the English longitudinal study of aging that's conducted in the UK. One of the significance of this article is that it contributed longitudinal evidence collected from a large cohort across 10 years with follow-ups every 2 years. While they also collected a wide range of data, this article's analysis was focused on age, physical activity, sleep duration, and cognitive function. More specifically, participants were categorized into 3 age groups: 50s, 60s, and 70s. Participants were differentiated into high or low physical activity group based on their reported intensity and frequency of physical activities. The authors considered the optimal amount of sleep to be 6 to 8 hours, and any participants having less or more sleep to be short or long sleepers respectively. Cognitive function, as the outcome of interest, was represented by a composite score based on the participants' performance on episodic memory task and verbal fluency task. Now, let's get into the findings of this study. The study found that at the beginning of the study, the average cognitive performance was better in the high physical activity group as compared to the low physical activity group. Further, comparing to their counterparts in the low physical activity group, participants with high physical activity was associated with a baseline cognitive benefit that increased with age. In other words, the older you are, the more you benefit from having more physical activity. On the other hand, looking at sleep durations, compared with short or long sleep, optimal sleep was associated with a baseline cognitive benefit for all ages. And similar to the trend observed in the high physical activity group, the difference between baseline cognitive performance for optimal sleepers and long sleepers increased with age. At the end of the study, the results shows that there were negligible differences in 10-year cognitive decline between high PA and low PA group. However, subjects in their 50s who experienced short sleep had a greater cognitive decline compared to optimal sleepers, and no significant difference in cognition were found between optimal and long sleepers. Taken together, these results suggest that while having high physical activity provided cognitive benefits, it does not necessarily reduce one's rate of cognitive decline. Having suboptimal sleep duration, on the other hand, could become a risk factor that accelerates one's rate of cognitive decline for those in their 50s. Next, let's have a look at the joint association of physical activity and sleep duration. As one might have expected, optimal sleepers with high PA had the best cognitive performance. They performed significantly better than participants in the low PA group, regardless of their sleep duration. And while little difference was found comparing with other sleepers in the high PA group, the authors suggested that the supposed cognitive deficit associated with long or short sleepers might have been compensated by the cognitive benefit one would gain from having high physical activity. Comparing longitudinal data, the study team had some surprising findings. Long sleepers with high PA had a less rapid cognitive decline than optimal sleepers, as indicated by the reference group. Short sleepers, on the other hand, had a more rapid cognitive decline than the reference group. In fact, the cognitive decline was so great that these short sleepers actually performed similarly to their low PA counterparts on cognitive tasks. This is particularly significant because if we recall the results at baseline, short sleepers and optimal sleepers with high PA were performing similarly. Hence, this would suggest that high physical activity was insufficient to ameliorate the rapid cognitive decline associated with short sleep. To sum it up, here are the key findings. More frequent, high-intensity physical activity and optimal sleep duration were independently associated with baseline cognitive performance. Short sleep was also associated with a faster rate of cognitive decline. Evidence of a joint effect of physical activity and sleep duration, along with the cognitive trend, also suggests that combinations of physical activity and sleep duration interact to influence cognitive scores up to 10 years later. The high PA group generally had the highest baseline cognitive scores regardless of sleep duration. However, for ages 50 and 60 years with short sleep, they are likely to experience cognitive decline within the next 10 years, such that cognitive scores of high PA groups are similar to those in the low PA groups. And that is all for now. Thank you for tuning in.
Thanks a lot. I think those studies are very important because, too often we think of sleep and exercise as important for healthy aging, and too many of the studies tend to focus on just one of these factors, and the complex interrelationships between things like sleep, exercise, diet, stress are very important for aging, and they're not studied together enough in these kinds of research. So I think that's a very informative research study there. It's great to introduce Stuart Cook. Stuart grew up in Kenya, studied medicine at St. Bartholomew's Hospital in London, did a PhD at the National Heart and Lung Institute in the UK, and then a postdoc at Harvard. He's a group leader at the MRC London Institute of Medical Sciences and a professor at Imperial College, and also at Duke-NUS here in Singapore. He was founding director of the National Heart Research Institute in Singapore, and he's been here for a while doing some very exciting research. His work has been focused on genetic heart muscle disease, and he's led to a number of new findings, including new diagnostic tests to look at inherited cardiac conditions. But more recently, he's focused on interleukin-11 and its role in a wide range of age-related conditions and healthspan directly. So the topic of his talk tonight is interleukin-11 as a therapeutic target for mammalian healthspan. Stuart, thanks for coming on the show.
Great, Brian, thanks very much. Thanks for having me. Looking forward to sharing with you over the next 25 minutes some of the recent data. So let's just get this share screen going. So let's just jump straight in. So a couple of my disclosures here, just for the record. So what I want to start by doing here is introducing Interleukin-11 to you, just very briefly, because not many people know much about this cytokine. It's a little studied cytokine. It's a member of the IL-6 family of cytokines. And long story short, in spite of what the literature will tell you from the earlier years, IL-11 is a pro-fibrotic in fibroblasts and also actually in the epithelium. It's anti-regenerative across organs. That means it actually prevents repair mechanisms, particularly in the epithelium, which are important for in aging and other common diseases in younger people, the repair mechanisms which restore function, for instance, to your kidney and your liver. And it's also pro-inflammatory, particularly in the stroma. So the stroma, which comprises of fibroblasts and vascular smooth muscle cells, actually plays a very important role in inflammation. Although they're not inflammatory cells, they are very important for inflammation, and IL-11 is pro-inflammatory in the stroma and also epithelial cells. Oh, got a problem with advancing my slides. So when we came to first study IL-11, we were aware that it could signal via its known canonical pathway, STAT. So this is IL-11 binding to its receptor, signaling via GP130 through STAT. And it was known a little bit that it also signaled through ERK. But what we've come to understand in a more detailed fashion over the last few years is in this pathway, the ERK, which then phosphorylates P90 risk, this can then phosphorylate LKB1, a very important kinase, on two positions, which inactivates it. So this has knock-on effects for AMPK, causing its inactivation, which can then activate TOR downstream and downstream TOR signaling. And this we have observed across cell types, across epithelial cell types, across stromal cell types. Seems a very conserved and robust pathway. And if you'd like to understand more about this publication, which came out just last year, I refer you to this the screenshot here, and the PubMed ID is just there in the bottom right. So if you'd like to learn more about that, this explains how IL-11 really strongly controls this module of ERK-P90 risk, LKB1 AMPK, and activates mTOR. So if you activate an IL-11, you activate mTOR with this sandwich of signaling in the middle. So why is that important for aging? Well, I know this community will know a lot more about this than I did a while back, but I came to understand that studies over many decades now have looked at the pathways, the key pathways involved in aging. And of these, some of the most important are MEK-ERK, LKB1 AMPK, and mTOR. And obviously mTOR being key here, target of rapamycin, which is probably one of the best studied aging pathways for lifespan that there is. So if I now overlay the IL-11 signaling pathway for you, you can then immediately see that why this might be irrelevant, because IL-11 is controlling these key components, at least in the studies that we did earlier on in cell types. So that opens the question, could IL-11 be important for aging, inflammaging? Everyone is probably aware that inflammation is a very important component of the pathobiology of aging, one of the hallmarks. And IL-6, which is a brother of IL-11, is actually quite strongly implicated as an inflammaging factor. So the question that rose to us is, could IL-11 itself be an inflammaging factor and contribute towards the diseases of aging, reducing health span and or lifespan? So obviously, to be important for aging, it has to be upregulated in aging. So what I'm showing you here are extracts from liver, and these are mice which are 12 weeks, 25 weeks, 50 weeks, 75 weeks, or 110 weeks old. And what we've simply done here is blot for IL-11. So this is a Western blot for IL-11. And you can see as mice age, as they hit about 50 weeks, you're beginning to detect an upregulation of IL-11, and that is then progressive as those mice age. So what's interesting then, if you look downstream of that at the IL-11 pathway, you see a correlative increase in ERK, an increase in P90 risk phosphorylation of LKB1. LKB1, this inactivates LKB1. a dephosphorylation of AMPK, which is consequent here. So you're inactivating AMPK. And you can see that during this period, there is a sequential increase in the activation of TOR and its phosphorylation of its downstream substrates. Excuse me. I'll decline that call. So this is correlative. IL-11 is going up in aging, and you have activation of those pathways in the liver and other organs. So I won't go into this detail, but just now showing you male and female mice here, either 12 weeks old or 110 weeks old. And in each organ that you look at, choose your favorite organ, say it's kidney, you can see that IL-11 is upregulated in male mice, old male mice, and also upregulated in old female mice. And that's true across all organs that we have studied to date, including the brain, which isn't shown here. A nice reagent that we have, and we generated some years ago, is an IL-11 EGFP knock-in mouse. So this is where we've knocked EGFP into the IL-11 locus. And this enables us to visualize which cells within the tissues are actually expressing IL-11. So if you look in young mice, there's no EGFP because IL-11 isn't expressed. But if you look into the older mice, you can then determine which tissues and which cells within those tissues are expressing IL-11. So this is liver. And what we're staining here is a transporter, which you find on the apical and basal side of the hepatocytes. And then you can see here in green, this is the EGFP signal, which tells you that this is where IL-11 is being produced because the EGFP is cleaved and retained within the cell. So in the liver, we can see you have high upregulation of IL-11 in hepatocytes. It's not the same story everywhere. So this is muscle. Obviously, sarcopenia is a very big problem in aging. And in the muscle, you can see here in the green that you're getting high expression, and obviously here in the vessel. So this is endothelial cell staining, and we also see expression of GFP in these structures here and here, which are nerve bundles. So actually in the muscle, you're seeing quite high expression in nerve bundles, peripheral nerve bundles, and the epithelium of the vessels, so different to what we've seen in the hepatocytes. And this is adipose cells. So here we're seeing high expression of IL-11 actually in adipose cells as we age. So this is a nice resource that we can pick out the different cell types and the different tissues. So what about, so I showed you a correlative relationship. What about a mechanistic relationship? So here we're looking at either young or old mice, and they're either wild type, 3 of them, or IL-11 knockout, 3 of them. And if we just focus now on the old mice here, you can see if we take away IL-11 that you inhibit P90 risk, you rescue the activity of LKB1 and AMPK, you inhibit TOR, its downstream substrate phosphorylation to orders down from it, and then senescence markers. So what this tells you is that IL-11 upregulation in age is associated with activation of this pathway, in particular of TOR, and also is associated with an increase in senescence markers across tissues. This gives you now a mechanistic relationship between IL-11 aging pathways and senescence across tissues. So we then began to look into health span and features that are associated with aging. So for instance, here we're looking at adiposity. So these are young mice here, and these are old mice here. These are IL-11 knockouts. I believe these are females. So as mice age, you get an increase in fat mass. So just looking at these dots here, you can see young mice become fatter. But if you look at the knockout mice, you have an attenuation of that increase in adiposity. And here you have lean mass, which is lost. And here you have retention of lean mass as well. So you're getting less fat, but retaining your muscle. And then if at the euthanasia, we collect the tissues, you can see it will actually weigh the tissues. So this is organ weight, and this is visceral white adipose tissue. Young mice put on a lot of white adipose tissue. And here that you're seeing a lot less of that in the knockouts. And that's also true for subcutaneous fat. So you can see that the loss of function in IL-11 is actually preventing the onset of aging-associated adiposity by different methods of assessing it. This is another metabolic disease that we all suffer from as we age, which is steatosis of the liver. So here in young mice, as they become old, wild type, you can see you have an increase in the fat content of your liver. And this is attenuated once again if you are a mouse lacking IL-11 signaling. And this holds true for other parameters such as cholesterol here. Again, cholesterol goes up in wild type, less so in the knockout. and again for serum triglycerides. So I think you can see here on multiple levels, we have improvement in the metabolic health of mice as they age lacking IL-11 signaling. We looked at biomarkers, aging biomarkers, and this is one component of that. This is a telomere length. And so if you look in wild-type mice as they age, you lose telomere lengths in your liver, your fat, and in your muscle. You do also lose telomere length if you're an IL-11 knockout mouse, but to a lesser and significantly retained extent. So lacking IL-11 improves metabolic function and is associated with reduced markers, biomarkers of aging here, telomere, and also mitochondrial copy number and function. Quite useful sometimes, I think, to see these things visually. So this is showing you IL-11 knockout mice here on the left-hand side, and these are wild-type mice, and these mice are brothers, all around 90 weeks old. And you can just see by looking into the cage that the knockout mice are leaner, more active, their coat color looks better compared to their brothers who are the wild types. And just here's a static example of sisters. So again, here the old mice are both the same age, with the wild type shown on the left and the knockout shown on the right. Important aspect of aging more holistically to assess is frailty, which is something very important clinically. And there are different ways of scoring this. This is a score that we have used in the lab, which is a 21-point frailty score, which we can then score various things from tremor to tumors to cataracts to hearing, etc. So if we then look at frailty in mice, we can look in, again, once young mice and old mice, and here we're looking at females on the left and males on the right. And if you're a knockout mouse, basically you have a much lower increase in your frailty score as you age compared to the wild type mouse. And that's true in both males and females. So a reduction in this holistic measure of whole body aging frailty, which predicts clinical outcomes. So that's all very well, but what about a therapeutic intervention rather than a genetic global loss of function? And so what we're going to show you, I'll show you here to end with, is an assessment of mouse aging, health span, across a period of 75 to 100 weeks of age, where they were given anti-IL-11 therapy once every 3 weeks, as shown here. So what's quite interesting with this is actually we can reverse some age-related phenotypes. So we can see here the mice will start with the body weight here normalized to themselves at 100%, but those getting anti-IL-11 actually lose weight. They're losing about 15% of their body weight compared to those that are untreated or getting IgG by the end of the time course here. These mice actually eat more, but these mice here getting anti-IL-11 are actually eating a little bit more, but they are losing weight. So what if we actually look at the fat itself? So this is epididymal white fat. So 75-week-old mice have this much fat. Untreated mice at 100 weeks have this. The placebo has this, and the anti-IL-11 has this. So you've had reversal in the weight of your epididymal white fat, adipose tissue, by the anti-IL-11. And actually here we're looking at gastrocnemius weight. So this is a readout of sarcopenia. So if you're a 75-week-old, you have this much muscle, you lose muscle if you're untreated or get IgG, but that muscle loss is less apparent if you're getting anti-IL-11. There's other functions that we can measure where we see a reversal. And so this is a glucose tolerance test and an insulin tolerance test. So 75-week-old mice, this is your glucose tolerance test. It's the same with placebo at 100 week, but actually you've reversed it from here to here if you're getting anti-IL-11 and 100-week-old mice compared to 75-week-old mice. And that's also true for insulin tolerance tests. That tells you better glucose homeostasis, better glucose handling if you get anti-IL-11 therapy. And this is now showing a reversal of muscle dysfunction. So these are 75-week-old mice, and this is full body strength, normalized to body weight. No treatment, it's pretty much the same at 100 weeks. Placebo, pretty much the same at 100 weeks, but anti-IL-11, you're actually getting an improvement, a reversal in your sarcopenic muscle dysfunction global readout. And if we measure frailty in itself, so these are mice normalized to themselves. So untreated mice progress in their frailty scores. Those getting IgG progress in their frailty scores, but those getting anti-IL-11 over this 25-week period of therapy do not progress in their frailty scores, which is quite remarkable, I think. There's a lot of interest in biomarkers of aging. I've shown you two already, the telomeres and the mitochondria. I'm showing you here the epigenetic clock. This is an imperfect measure in the mouse. These mice are actually all 100 weeks old. So these wild type, these here getting the IgG should be actually here at 100, but we're at the top end of the range where we can measure this with this particular assay. But that notwithstanding, so those getting IgG are estimated to be about 80 weeks old, but those getting anti-I11 are estimated to be almost 50 weeks younger. And that there's another clock, different epigenetic markers in the muscle gastrocnemius. Again, those getting IgG estimated to be about 80 weeks old, those getting anti-I11 about 30 to 40 weeks younger by the epigenetic clock analysis. Just a little bit to end here about the I11 signaling in adipose tissue. So what we find if we profile the adipose tissue is that those getting IgG, you have undetectable UCP1, uncoupling protein 1, but those getting anti-IL-11 have a large upregulation of UCP1. And actually what you find in these mice getting anti-IL-11 is this reactivation of a Bayesian browning program in the fat, which is actually beneficial, very metabolically beneficial. And I didn't know this, but loss of white adipose tissue Bayesian is an aging phenotype in humans and mice. So as we get older, we lose our ability to have thermogenic adipose tissue, which is a way of burning calories and keeping your weight off. And what we find is anti-IL-11 actually reactivates this age-repressed program of white adipose tissue Bayesian. And if you look then at the protein level, this is 75-week-old mice. They don't have UCP1. If they get IgG to 100 weeks, they don't have UCP1, but you see it turned on again with anti-IL-11. And here is PGC1, which is important for mitochondrial biogenesis. You have it in 75-week-old mice. It's reduced with a placebo at 100-week, and it's restored over and above the 75-week-old level with anti-IL-11 therapy. And actually, if you look back at these mice now in both males and females, the IL-11 receptor knockout, the IL-11 knockout, or those either getting IgG or anti-IL-11, you have an increase in body temperature with loss of IL-11 function, which is a global readout that these mice are burning calories through a futile cycle with UCP1 upregulation in their fat. If you're interested, this phenotype is actually very, very similar to that seen with a white adipose-specific raptor-deleted mouse, and that is specifically through its activity on mTOR. So we believe that what the phenotype that we're seeing here is that when you inhibit IL-11, you prevent the activation of mTOR in a raptor way, and we actually recapitulate this phenotype of UCP1 upregulation and a lean mouse through this pathway in aging. So ending now, talking about therapies for lifespan. So these are some of our most favorite pathways in the aging field that people are trying to target with trametinib. Is toxic, metformin is not very nice. Rapamycin works, but dose-limiting toxicity, and senolytics also with significant toxicities. So we would like to make the argument that anti-IL-11 gives you a combination therapy because you inhibit ERT, you activate LKB1 AMPK, you inhibit TOR, and I've shown you that you diminish senescence markers throughout tissues, and I could show you a lot more data on that, but IL-11 is actually very important for the senescence of epithelial and other cells. So we believe this will have a good safety profile and limited on-target toxicities, as you're only inhibiting these pathways which are downstream of IL-11. So normal ERK activity, normal mTOR activity is still possible. And these are human knockouts who are pretty normal apart from a mild abnormality of the skull during development. So in terms of anti-IL-11, so we published this manuscript in Nature 2017 when we found IL-11 was important for fibrosis. There are 3 clinical trials now in progress for anti-IL-11. This one here actually just reported its phase 1 safety data at the European Respiratory Society, I think last week, showing very nice safety data of high-dose anti-IL-11 receptor therapy. So again, giving good thought that this could be a safe and effective treatment in patients for long-term therapy in aging, which obviously is a consideration. And to mention here, under my disclosures, we have a new company here in Singapore called VVB, where we've been generating specific antibodies which are super potent that we hope to use for aging trials, which can be given via an administrative route and in a way that is conducive for long-term therapy out of hospital. So I'd like to thank the team, led very ably here in the middle by Anissa at Duke-NUS, but the rest of the team also playing a very large role in this study, which has been over 6 years long. And with that, I will stop and happy to take questions.
Thanks, Stuart. Data looks really exciting.
Thank you.
Have you done? I don't think you've done this yet. Maybe I'm forgotten. Have you done survival studies with the IL-11 antibodies yet?
Yes, so those obviously take a little bit longer, but they are ongoing and very exciting.
Yeah, I think that'll be, I mean, everything looks great. I'd be surprised with all those phenotypes if the animals don't live longer, actually. So be interesting to see. Yeah. I think, one of the things that we point out when we talk about TOR a lot, and a lot of people don't pay attention to this, is that that people think of TOR inhibition as extending lifespan, and they don't think about the fact that mTOR signaling is naturally going up with aging, and that what rapamycin may be doing is not inhibiting TOR to below some baseline level, but to rather restoring the dynamic regulation of mTOR with age. It seems like you think that maybe the driver of activated mTOR is IL-11 based on the fact that it's going up with age. And I'm wondering, it seems like that's pretty broadly across tissues, which is maybe a little surprising because mTOR is regulated somewhat differently, for instance, in brain and muscle and liver. So I don't know how broadly you've looked at that and what your thoughts.
So I think upregulation and mTOR activation in age it's sequential, and you can see almost like a dose dependency between IL-11 levels and the TOR activation. So I do believe that the increase in TOR activity with age is strongly linked with IL-11 upregulation, and that's the pathogenic TOR signaling. But below that, you have physiological TOR signaling, which is still needed. The difference we see across tissues, we haven't done head-to-head, but there is a difference in the level of TOR activation and IL-11 upregulation and the pathways. It's just we haven't done those head-to-head on a Western blot, which you'd need to do side by side to be able to appreciate that.
Yeah. How much of this is through senescence with IL-11? That I always get stuck on the same questions with inflammaging, and I think the audience is probably tired of hearing me ask this by now. But there are many different sources of inflammation during aging, and senescent cells are one of them. IL-11 seems to be affecting at least the SASP through mTOR and maybe senescence in general. Are there other pathways to inflammation from IL-11, or do you think it's all going through senescence?
No, I think it's more than just senescence. And I think that's why it comes back to the IL-11 effect on things such as repair and regeneration. So it directly inhibits the epithelial ability. It pushes the cells into a mesenchymal state, so they're no longer able to proliferate, regenerate, and restore. So I think that's an important factor. Fibrosis is another factor. It's directly activating fibrosis, which disrupts tissue function. And I think, and obviously IL-11 is a component of the SASP, and actually, and it drives senescence within different cells. And that's been shown by a number of groups, including our own, which is an mTOR-dependent senescence program in different cell types, including epithelial cells and fibroblasts.
I was wondering about you were looking in the muscle and you saw IL-11 going up in vessels, I think, but also in nerve bundles. What about neuromuscular junctions? I feel like that's an area that's not studied as much as it should in the context of aging. So both in terms of the nerves and also the nuclei and the myotubes that are associated with the nerves.
Yeah, I think that's a really important question. And again, I think the nice thing about the IL-11 EGFP mouse is we can see where it is, and we see a super strong signal in the scaly muscle, in the nerve bundle. The NMJ junction, we've been trying to look at that, and we have ongoing staining to try and assess that more fully. My personal guess is that IL-11 in the muscle is causing denervation, or at least dysfunction of innervation. And then when you inhibit IL-11, you're restoring function, because we see restoration of muscle function over and above restoration of muscle mass. So that speaks to me that it's not just muscle mass that's getting better, but actually the functional units of the sarcomere, which speaks to me about innovation.
Yeah, one of our faculty here has been, Sheen Tsai, has been trying to sort out the relative roles of components downstream of mTOR and age-related muscle decline. And 4-ABP has some roles and S6 kinase has others, and muscle function and muscle mass are not always together on that with aging. So there might be some interesting comparisons there.
Yeah, and I think there's been some people who have actually localized mTOR activation within the skeletal muscle to the NMJ. So I think there's already data out there that the mTOR activity is acting in the nerves.
Mm-hmm. One thing that strikes me is that when you look at like your studies on lean mass and the IL-11 knockout and adipose tissue and cholesterol, many of these phenotypes, there's clearly an effect with aging, but it looks like there's a smaller but significant effect in the young animals too. That doesn't take away from the importance in aging, but it also makes you wonder if there's a developmental or early adulthood phenotype as well associated with IL-11 knockout.
Yeah, so for sure, in some and you have to look carefully, and some phenotypes are more apparent than others, but you can see it in the signaling. For instance, AMPK, you can see it in quite young animals, and certainly, I mean, we see a big hit when animals are turning about between 30 and 40 weeks, and that's when we think IL-11 is really coming on. But even in the younger animals at sort of 15 weeks, you can see a small effect. Likely because, well, we don't believe IL-11 comes on in health, and certainly lacking IL-11 helps you live healthier. But there is a signal there in youth as well, in younger animals as well, for sure.
Yeah, I wonder how much is adipose related. You know, these animals are gaining weight pretty rapidly. Pretty standard result from aging mice. You know, Jim Kirkland and others have shown a lot of senescence in adipose tissue. You know,
I think so.
And like on a calorie-restricted diet, would that minimize the phenotypes, or would a high-fat diet exacerbate the phenotypes?
Yeah, so in a high-fat diet, IL-11 is very largely upregulated in the fat and the liver. So, and we haven't looked at calorie-restricted, but basically when you fat load a cell, a hepatocyte or an adipocyte, they start to make IL-11. So IL-11 is secreted in response to that. So there may be a link between calorie restriction and IL-11 more directly in aging. That actually, and just to speak to your point, is it all the fat? Well, it isn't. So we see very strong effects in the kidney, unpublished, not really related to adiposity. And I think when you see the effects happening in nerve bundles, again, that speaks to a bigger effect beyond just fat, also in the brain. So I think the fat is a strong phenotype, but I think there's organ level phenotypes which are independent of that.
Well, that was one of my other questions, I think, which was, you know, you see phenotypes in a whole range of different tissues, which is, you know, common with things that slow aging. Where's the primary effect? You know, is IL-11 doing something in every tissue, or are there key tissues that have knock-on effect on other tissues, and which leads to the question of tissue-specific knockouts? So.
Tissue-specific knockouts in aging, yes. So I mean, I think they're all good questions. I think in the mouse, there's definitely a lot of activity in the adipose tissue. And I think that would be a good place to look. You know, obviously the Browning and Beijing phenotypes is quite a mouse-driven phenotype. One has to be a little bit careful of that. I think in humans, we might see it being driven more strongly elsewhere. It is upregulated in humans. In aging, you can detect it in the serum in very old individuals. And I think, you know, in humans, maybe you might get a bigger hit in the kidney, for instance, which is a classical aging organ in the humans. But I do think you might also see a fat phenotype in the humans if you can restore some Beijing to that. So I think it's an open question, but I do think there is organ-specific effects, and you can see that in the cell types expressing IL-11. It's not just the fat.
I mean, if you look at it through the lens of mTOR, you would, you know, tissues where mTOR inhibition might be really important. Kidney certainly comes to mind. I mean, it's used clinically, rapamycin and rapalogs are used clinically in that context. Also, there's a lot of data on cardiac function with aging and improvement with rapamycin. And so I have you, most of that's preclinical, but I always wonder why there aren't more studies with mTOR inhibitors and cardiac hypertrophy, dilated cardiomyopathy, and whether IL-11 has an involvement in that as well.
Yeah, so we've looked a little bit at the heart. I mean, the thing about the aging heart and the C57 black 6, it's very hard to find a phenotype. That's the trouble. So we've looked more at molecular level of readouts of stress, such as BNP, which is a natriuretic peptide, and we do see an effect there. So we don't have it as organ level physiology, but we have it at organ level molecular readouts. And I think there's one thing to add about I-11 that people forget, and it's probably the way I propose it as well, the mTOR axis, that is very important, but let's not forget that it has the whole STAT3 inflammatory axis as well. So what we would propose at I-11 over and above what you get with TOR inhibition is you get more of that inflammatory component because you hit the STAT pathway directly.
A kind of a strange question, but when I looked at your frailty changes at the individual mouse level, most of the mice don't have increases in frailty, but there are a few that do with the IL-11 inhibition. And one of the things we're thinking about a lot now is sort of personalized aging mechanisms. And so I'm wondering if you look in the mice that still have an increase in frailty, is there some correlation with body weight in those mice? I mean, not all mice gain weight. And maybe IL-11 inhibition is a problem if the mice are not overweight, or some other way to separate the mice that respond from the mice that don't respond to IL-11.
Yeah, I think that's obviously, as with aging phenotypes in mice, the older you get, the more diverse your phenotypes get. I mean, there's a huge spread. And actually what we do see with anti-11 is it reduces it really reduces your variance in all of the phenotypes we measure. It brings it closer together. But I think it's an interesting question. It could also be related to the biological age of the mouse. When you start the intervention, they could actually, although they're all started at 75 weeks, some of them could actually be, I don't know, 85 or 100 weeks old by the clock, whereas others aren't. So I think one could think about stratification or looking into that rather than body weight itself. I don't believe we see much relation to just body weight.
Yeah, I kind of just threw that out as an example, but we don't tend to do we don't even tend to show data like that often in mouse studies. And I think it's really important because there's going to be non-responders in any longevity intervention. And we have to start thinking about, there's so many clinics that are promising personalized aging, and I don't know if we can do that in a mouse right now. So I'm always interested when you see differential responses and things.
Yeah, for sure. There's outliers always. And I think also scientifically, I think it's important to show that.
So when I come to the translational component, you've really chosen to go down an antibody path, it sounds like. Why that rather than small molecules? Is it practical, or what's motivating this approach?
Yeah, so I think in terms of specificity and dosing, I mean, if you take the example of Humira anti-TNF, that's been around a very long time. People have tried to generate a small molecule inhibitor that's better than it. It's a very hard thing to do because antibodies work so very well, especially against secreted factors. So I think there's a nice example out there with this anti-rank ligand antibody, which is used to treat osteoporosis in older people. My mum gets it. And you get a subcutaneous dose out of hospital once every 6 months. Mm-hmm. That's a pretty nice dosing regimen to potentially think about. And I think with a little bit of effort, that could be done with an anti-IL-11 therapy in the clinic.
Yeah, and I'd forgotten what you did in the mice, but I think it was fairly consistent antibody treatment. Or can you do it more sporadically?
Yes. So we went. So this is a murine anti-IL-11 antibody that we have in the lab. It has a half-life in the mouse of about a week. So that's a pretty standard thing. Antibodies in humans have a half-life of about 28 days, and you can do other things to try and improve that. So, and there's examples out there, anti-rank ligand antibody, which is given every 6 months, because it also depends on the expression of the target, obviously.
Also, you may not need to have the antibody there all the time, and if you're reducing senescence. So that's
you could even imagine you might even be able to have a pulse clearance, and then a period when you don't need the antibody at all.
So just one more thought, and then I'll go to the questions. You briefly showed humans with IL-11 mutations. Can you comment more on those? And I'm wondering and also from a therapeutic perspective, I'm wondering if there are any adverse things to worry about from inhibiting IL-11.
Yeah, so in the studies that we've done in the knockouts and also the ongoing lifespan studies, we have seen no detrimental effects on anything, only benefit. My premise is that IL-11 only comes on in disease in the adult, and I think that's beginning to hold true. In development, in some individuals, if you have no IL-11 signaling at all, you can have a craniosynostosis where the fusion of the plates don't fit quite so well, and a tooth eruption disorder, and a maxillary disorder, and conductive disorders of the ear. And so it's a developmental bone defect, which is mild and incompletely penetrant. And so that's what you get. If you inhibit IL-11 in the adult, will you have any adverse effects? I think it's always possible, but the data today is not showing a lot of that. In fact, Lassen Therapeutics have an anti-IL-11 receptor antibody in the clinic, just finished their phase 1, no safety issues reported. So one has to be careful when one goes super long term if that's going to be a lifelong therapy. But I think of all the targets out there for aging right now, this is probably one with a better safety profile.
Yeah, and it takes advantage. You know, people talk about antagonistic pleiotropy. You know, and you know, genes and proteins are around for a purpose. But if there's a developmental role for that gene that's not needed in the adult, then there's a temporal window that you can take advantage of to target for aging. And so
That's exactly right.
It seems like this might be a candidate for that.
Exactly that.
Cool. Well, you know, I could keep asking questions forever, but I'm losing weight here, sweating, and maybe it's better to bring in Kalisha to ask you some questions from the audience.
Hi, Professor Stewart. Thank you for that presentation.
Welcome.
I have some questions here from the audience. One of them builds upon Professor Bryan's last question. Will the use of anti-IL-11 as a therapy potentially affect the host immunity and its ability to fight infections?
Okay, so I'm going to answer that directly. So I think the answer is no, and I think the evidence can be pointed to from human genetics, for instance. So if you lack the IL-6 protein, IL-6 receptor, which is a brother of IL-11, you get all sorts of infections. You get pneumonias, you have eosinophilia, you end up with abscesses, etc. Because that is important for immune cell function. IL-11 IL-11 receptor is not important for immune cell function, and humans who are knockouts don't get infections. They don't have any difference in their platelet counts, etc. Nor do mice, given the therapies. And actually, as I just said, the phase 1 trial just reported by Lassen Therapeutics, they've been given this to humans now at high dose. Haven't seen anything.
Okay, all right, thank you. The next question asks, since the IL-11 receptor is highly expressed in the gastrointestinal tract, is there any effect on the digestive system? And could this be the mechanism in which we see the decreased weight gain?
So the decreased weight gain, I think, is fairly conclusively due to white fat beiging and browning, and that's why you have the increased temperature, and we've done this in metabolic cages. So I think one can, I don't think to exclude, when we're actually doing another experiment of measuring the calorific content of mouse poo right now, just for a reviewer. But I think the weight of evidence is, you know, you have huge amounts of beiging, you have an increase in temperature, and you have an increase in the burn of the fat. So that, rather than a gastrointestinal loss problem. You do see IL-11 upregulated in some diseases of the bowel, such as Peutz-Jeghers syndrome and also colitis. And actually, IL-11 is thought to be a target for bowel disease. So the other way around, really.
Okay, all right, thank you.
Have you looked at barrier function?
We haven't looked at barrier function because we don't know how, but we'd love to do that because I'm going to guess it's going to be improved. We haven't looked at it, but if you know how, tell us.
Yeah, we can talk.
Okay, our next question is, what are the beneficial functions of IL-11 in normal non-aging physiology?
Sorry, that's all?
What are the beneficial functions of IL-11 in normal non-aging physiology?
Yeah, so
why is IL-11 there?
Yeah, so again, so in humans, IL-11 has actually accumulated neutral genetic loss of function variation. So basically, there is no selective pressure within adult humans to maintain IL-11 function. So there's the very strong human genetic data to show you that. What I-11 is important for is important in the fishes for regenerating their tails. It activates a super strong mesenchymal program if you want to regenerate your tail. So humans don't regenerate tails or arms, and we can have children without having to retain a regenerative function. So although it has a role in a small way, incompletely and variably in development of the skull and a little bit of your bone length, in the adults to date, we have not seen a physiological function, only a disease function.
Okay.
That actually raises a question though around wound healing, because mTOR is activated during wound healing. Senescence is thought to play some role in wound healing. It might suggest that IL-11 has a positive role in wound healing.
Again, I think that's a very... we were asked that question the other way around when we showed it was important for collagen function, but obviously we then did all the experiments, and certainly the wound healing on a just a global level is normal. Whether it's improved, it might be, and there's certainly a lot of evidence for activity of GP130 signaling in general in diabetic ulcers and failure of wound healing, and obviously IL-11 is a GP130 ligand.
Another question we have from our audience is there any relation between IL-11 and the anti-aging protein klotho?
So that is a question I do not know the answer to directly, but what we have looked at is a relationship between IL-11 and IGF-1, and there is a direct relationship between IL-11 activity and IGF-1 secretion from fibroblasts. So and obviously IGF-1 is another well-known aging factor, and we think IL-11 is directly or at least importantly involved in the regulation of IGF-1 levels.
Thank you for your answer. Now, I have one last question from myself personally, because I focus more on the liver aging.
Now you're in trouble.
So you've seen a reduction in liver weight once you knock out IL-11. Do you think IL-11 therapies could be used to reduce non-alcoholic steatohepatitis and NAFLD, non-alcoholic fatty liver disease, and the following fibrosis of the liver?
Yeah, so when in our initial studies that we undertook at Duke-NUS back in the 2018-2019, we showed that basically IL-11 is critically important for both NASH and NAFLD through its activity in hepatocytes. And if you lipid load a hepatocyte, it will secrete IL-11, causing hepatocyte dysfunction, but then also speak to the stroma to activate hepatic stellate cells to cause fibrosis. So it's important in NAFLD and NASH. And actually, one of the stated intents of Boehringer Ingelheim, who bought one of our drugs, is in NASH. So they are looking to do phase 2 clinical trials at some stage in the future in NASH, and that's a stated intent of them. And actually, there's been a couple of papers come out just in the last few weeks by a couple of other groups showing that using siRNA approaches, you can treat either CCL4-induced liver fibrosis or NASH, independent of our studies.
Yeah, that raises a question for me too, I think, which is, with these therapeutic antibodies, obviously you're interested in looking at aging directly, and I think that's great. I never want to discourage anyone from doing that. But from the company perspective, are you looking at disease indications or?
Well, I think we've discussed this before. I think sarcopenia is an area of great interest because we see very marked effect on muscle function, and it's kind of a bit independent of the obesity functions. One can argue the case there, but it is, I think, independent. So I think sarcopenia in aging is an area that could be particularly interesting for an anti-IL-11 therapy, and something I would actively like to explore. I think just to come back to one point I mentioned about regeneration and how IL-11 is important for regeneration in the fish. I just want to add that senescence is also very important for regeneration in the fish. So, and I'm wondering if IL-11 is not only activating this mesenchymal program that we see, but also activating senescence in the fish tail as it regenerates its tail, because senescence is actually also turning out to be important for regeneration in regenerative species. So I wonder if there's a bigger picture here where you can connect a few more of the dots.
Yeah, it would be interesting if there was a way to look at IL-11 inhibition in killifish. We've got that model up and running now, and we love it because these fish age really rapidly, and you can look at some of the different pathologies that are hard to look at in a mouse. So,
yeah. The thing with the fish, though, is its regeneration, the epimorphic regeneration of a fin or a limb requires the blastema, which is I-11 driven. So that is actually dependent on I-11. So it's almost like the antithesis where regeneration in the fish is
maybe.
Actually, we have one more question from the chat. They're asking, does downregulation of IL-11 enhance lifespan? Do you have any C. elegans experiments showing that or anything?
We have mouse experiments, but they're ongoing.
Okay, great. Thank you.
He's keeping it close to the vest, but he's got that smile that makes me wonder.
Yeah, you never know.
Stuart, thanks for joining the show. I look forward to talking more about this, and it's really exciting data. So thanks a lot.
Thanks for having me.
Felicia.
Thank you.
I want to remind everyone to use the chat function and panelists and all attendees option. Leave comments on the show. Remember, our Unlock Healthy Longevity Supplement Conference will be the February 29th and March 1st of next year. Registration's open. You can also sponsor part of the program if you would like to. It should be really exciting. I hope you can all come to that. Check out our website on a regular basis because we're announcing great international speakers as they sign up. Also, spread the word. October 5th is our 100th episode of this show. And I was a lot younger when we started these shows which was in November of 2020 during the pandemic. But unlike COVID-19, it stopped and we didn't. So it will be at our 100th show soon. The center is looking for postdoctoral fellows in geroscience, nutrition, supplements, and aging in general. So let us know if you're interested in joining in that form. The next episode will be very interesting and a slightly different view on aging. It'll be Jose Cordero, who, if you're in the longevity field, you know who he is. He just came out with a book, The Death of Death, The Scientific Possibility of Physical Immortality and Its Moral Defense. So that should be a really interesting conversation. We also have a video coming up, and I want to make a comment about that. One of the things we try to do on our webinar is to paint a very broad picture of different views on why aging happens and what people might do about it. This video is about why someone is spending millions of dollars to be 18 again. I don't actually advocate everything that's being done here, and I do believe in democratized longevity, but I also believe in informing everyone what's going on. So watch the video and decide for yourself.
The people who I admired the most did things typically over a multi-decade time frame. They surveyed the landscape of things that they can do, and they identified the thing that can barely be seen and say, I'm going to try to do that thing. At the age of 34, I thought it's time to try to do something meaningful on that kind of multi-decade timescale. My objective with Blueprint is to demonstrate aging escape velocity using the best science, trying to do all the appropriate interventions to neutralize my aging process. 1.8, so even better than last time. So I think my first one was 3.4. A few things have distinguished what I've done to date. As far as I know, I'm the only person that has publicly posted my data. So I'm not saying, here's my protocol and believe me. I'm saying, here's my protocol, here's all my data, and here's what I'm experimenting with next, and here's where I've made mistakes, and here's where I'm trying to fix it. It's really a stacked process of maybe 100 different things I do, because you have to think about the body in its entirety. 118, is that right? Mm-hmm. How the heart ages is different than how the lungs age, and how the lungs age is different than how the brain ages. You have to really think about it from a holistic perspective. So it's the common things like diet and exercise, but it's also a much broader consideration. So I have a team of doctors that I work with, and we just go through this rigorous process of measurement, gold standard science, implementation, measurement. Is produced near perfect health for me. No matter how extreme I've had to be, no matter how eccentric people perceive me to be, because I'm outside the norms, demonstrating that age can be arrested would change everything.
I'm feeling like sunshine, like springtime. like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.